Hey guys, we're back with my medical podcast, and today we're talking about nanotubes and how they might actually be facilitating cancer growth within your body. We've got self-proclaimed doctor and nanotube expert, Dr. Nanospert Nanotube. Today we're coming at you from my neighbor's basement, where they have locked their stepson away for multiple years at a time. He seems harmless. I'm sure, he'll be okay.
He doesn't lie.
Yeah. So, what is a nanotube?
Tunneling nanotubes are thin, hollow plasma, according to Google. They're plasma membrane projections that directly connect the lumen of one cell to the lumen of the other. And what these do is exchange intracellular material, including mitochondria, mRNA, and DNA of the cell. Up until recently, these bridges of cell communication have been seen as harmless forms of communication. However, researchers are speculating that these intercellular interactions are in fact facilitating cancer growth.
Okay, so are these efficient at exchanging material between cells?
Efficient? I would think that the polarity of the interior nanotubes is the same as the interior of the cell. So exchanging ions, organelles, and genetic material is going to be highly efficient, right? Organelles, for example, can't flow naturally through a medium different than their natural environment. So the nanotube serving as an extension of the inside of the cell to another cell helps allow the organelles to go past through.
Okay, cool.
Yeah.
So can viruses hijack the system and use it to their own advantage?
So it's widely known that viruses produce particles that contain all their DNA and RNA to spread the infection through human-to-human interactions and throughout the body, and these particles are used so that the viral information will be protected as well. Once a virus is in the cell, it's speculated that the virus is actually hijacking these naturally occurring systems of TNTs to get the genetic material information across more efficiently and without any threat of immunodetection. The viruses comes along and forces the cell to make more nanotubes more exaggerated than the cell normally does. They do this by replicating at low, undetectable levels. With this method With this method of infection, the virus doesn't have to be worrying about spending more information or energy to form particles, and will instead formulate these methods like manipulating nanotubes to their advantage. I feel like we can connect this to mechanisms to cancer cells, right? Cancer!
Hi, dude. So,
Hello.
Since you just killed my whole crew, you think that you could give me your name?
Yes, I am Trevor Jameson.
You think you can tell me how cancer uses nanotubes?
Yes, I actually know a lot about that matter. I actually know a lot about that information. So basically, think about this. There's a cancer cell and there's a somatic cell. So basically, the somatic cell is cancer cell. So this cancer cell starts building a nanotube to the somatic cell. And the cancer cell sends an electrical signal. So think about texting. You're texting someone, texting. And then basically the signal is being transduced through the nanotube. And we think about transduction basically amplify. Now the signal goes to the somatic cell and it's give me your mitochondria, give me your lysosomes. And basically those organelles go through the nanotube. And now it has freaking no organelles to do anything. Because mitochondria is ATP stuff. It produces ATP. And also lysosomes, this cell is going to be full of waste. It's not going to be able to get rid of it because it has no ATP or lysosomes. So basically, this somatic cell is going to die. And basically, this cancer cell is going to use all of that somatic cell through lysosomes by breaking down that matter through hydrolysis, which is using water to basically break down polymers to their monomers, and basically use that to grow and reproduce.
Okay. I like it.
Yes.
So, would you say that shear stress applied to nanotubes plays a role in this exchange?
Yes, I actually do believe that. I believe so. That is one of them. So, think about it like this. According to Brainly.org, it has been seen that mitochondria that undergo shear stress produce more ATP, which can contribute to more CO2 output. This makes sense because the cellular mechanics and physiology among the muscle cells, for instance, require movement to activate more energy production. Right? Mm-hmm. So for cancerous tumor development, the increase in CO2 is associated with it to achieve its hypoxic environment. The body is not equipped to handle this level of exchange between CO2 and O2 when it comes to cancer, as in it is not designed to deal with a tumor producing a microenvironment that happens the body ability to bring O2 to exchange it with CO2. Bruh. Something to note is that mRNA travels along these tubes as well. mRNA, and I speculate that these are undergoing a degree of shear stress that possibly degrades them. A degrading mRNA molecule might not have the right nucleotides to code for vital proteins, which would cause incorrect binding of peptide bonds and result dysfunctional proteins / mutations, if you will. So when the degraded mRNA molecule reaches the ribosome of the accepting cell, since not all peptides are synthesized properly, the resulting protein will not be correct to carry out certain functions.
Okay, cool. So that's how cancer harnesses the power of nanotubes to facilitate its growth.
Keep worshipping me.
Yeah, y'all heard it here.